Okay, we're live on Zoom now.
Okay, excellent. All right. Well, good evening, everyone, and welcome. Thank you for joining us for the March meeting of the Mecklenburg Audubon Society. I'm Malia Klein. I'm the president of Mecklenburg Audubon. And like we always do, Christine is going to start us off with a great slideshow that talks about what we've been doing, what we've been seeing, and where we're going from here. So, Christine.
Christine, are you still there?
I am.
Okay, first up, I'll just preview a little bit about what's going to be happening in about 10 minutes. We're going to hear an excellent presentation on bird hearing by one of our board members, Dr. Larry Leamy. Look forward to that.
Okay.
Christina.
Here's just another reminder for everybody. It's been a while since we let you know about the birds and beans. I believe the discount code is still working if you're ordering online and you get free shipping. So don't forget to get your coffee.
Good. I want some now. I don't have any. I wish I did. Okay, tonight we are honoring a cherished friend of Mecklenburg Audubon who passed away suddenly on February 20th. This amazing human being with the 1,000-watt smile was a pastor, a tireless community advocate, and he and his wife, Melissa Gaston, were our partners in creating the Druid Hills Birds, Nature, and Community Day Festival. He's shown here with Jill Palmer, who made this whole relationship come together, and they're at the inaugural event, which was so much fun when we launched that. We plan to continue this festival in Daryl Gaston's honor and to dedicate a new pollinator garden to his reverence for the beauty of nature. We'll miss you, Daryl.
Okay. And still in a similar note of advocacy, we have some of our own budding advocates here at Mecklenburg Audubon. As you guys know, the Audubon North Carolina every year does a state event they call Advocacy Day, and it gives a chance for some of our members across the state to speak directly with politicians in our area who are making decisions that may impact birds and wildlife and the things that we care about. So we really wanted to give some special thanks to those volunteers from our particular organization. Matt, Janet, Natasha, Noreen, Debbie, Richard, Christina, Dennis, Catherine, and Amy. We had a big turnout this year, 10 volunteers. Matthew Winthrow on our board, our advocacy chair, headed up this group, and we really had a great turnout. And being everything's virtual, they zoomed the day away, and we thank them for their efforts.
Excellent.
Hey, yes, thanks. Sorry, I'm not here by video, but I just want to give my appreciation to everybody who was able to join us. And I think it was really successful, and I look forward to doing it again next year.
Thank you, Matt.
Matthew, and I see John Altry up there. He's I think Janet Palmer, who's in that picture too. He's her representative. So he's a great guy.
Mm-hmm. Right. I noticed that he liked our webpage when I had posted about Advocacy Day, and we invited him to follow the page, and he does. So that's nice.
Good. Excellent.
Okay. Well, Natasha sent some pictures off to me this week when I was asking folks to send some of their local findings, and she was quite excited that she got a new camera recently and did not have to go far in her neighborhood to get these interesting shots. So the hooded mergansers are starting to do their little mating dance. The ladies don't seem all that impressed just yet, but but there it is. And I believe those redheads, both of those I think are found in just some local little pond in her neighborhood in South Charlotte. So I thought that was kind of neat. And the other thing is we're getting ready to start observing for the North Carolina Atlas, and we're going to be paying more attention to bird behavior and, excuse me, Oops,
So it looks
sorry, I
like,
hit a button.
so it looks
Um,
like Natasha's great blue heron is up there. I don't see any sign of a rookery or any
Yeah,
nest.
sorry. Yeah, so that was her comment to me was, I didn't realize they even go way high up in trees like that, which I thought was kind of neat. It is something you see every now and then.
Yeah,
A couple months ago, I had one on my neighbor's roof, which I thought was a little weird. And her close-up is really great. I love that little wisp of his head feather there.
nice.
Okay. This, apparently they're quite regular winter wrens on along 4 Mile Creek, which is the greenway nearest my house. And so I've been making a point to observe them more carefully this season, and I've been seeing them a lot. In fact, I saw him again just today, but he finally posed up nicely for some pictures last week. So that was that. And Gretchen had this really cool picture that she sent in. She is in York County in South Carolina, and she found this black vulture egg in an abandoned trailer somewhere nearby a neighborhood. Someone said they think her son noticed some activity and she wondered about it, that she thought it would be a good place for them. So she made a point to look and she spotted the egg, which is really cool.
Nice. That's gorgeous. Okay, so Christine is calling these really cool sightings because they are in Ohio, which is where our previous Mecklenburg Audubon president, Jim Guyton, is spending some time these days. And he has sent us these great pictures. I'll try to do them in order. So the short-eared owl on the bottom left, he saw that in Killdeer Plains National Wildlife Refuge, which is about an hour north of where Jim is currently in Columbus, Ohio. So that was extremely cool. He got some great shots of that. They're just,
Yeah, look at the feathered legs. It's really his
and those
talons
feet,
and his
those
feet.
puppy
Yeah.
feet just are amazing, amazing. I'll do the snowy owl, and then if you want to talk about the other two, Christine. So the snowy owl, which is just stunning, was at Alum Creek Reservoir. And Jim reports that she's very healthy and has been there working the dam at the reservoir ever since Thanksgiving. So she must really love being there and must be some good hunting.
Yeah, very cool. The Lapland longspur and the snow bunting, they were both seen at Ohio State University Farm.
And you got to say the Ohio State, Christine.
Yes, right. I noticed it's all in capitals. Oh, yes,
the Ohio State.
Okay, I got you. So that's right close to Jim's neighborhood when he's staying in Ohio, his hometown, when he's with his dad. So those are some winter species. I used to see them in New York occasionally, and really neat looking birds. And they travel in loose flocks, usually on fields, or like the snow bunting, it's like in a parking lot. They like gravel and grit and snow, and really pretty birds. I love a snow bunting. I think they're beautiful. Very elegant little sparrow-like birds.
Nice, very nice, beautiful pictures.
Okay, ta-da! Good news.
Just in time for spring migration, field trips are reinstated. We've really missed it. Judy was saying earlier that on her walk today, people were so excited to be out. She just couldn't get them to stop talking to each other and bird.
Yep. So Steve's going to go over the offerings in just a bit, but just for now, at least know we are back in business with all the usual safety precautions, of course, but we are ready to go just in time.
Yay, yay. All right, the Backyard Birding Challenge. Again, this was Patty Maston's idea. Christine, you are closer to the top than I am, so why don't you go through it? I'm somewhere way down there.
So first, let's take a look at Steve's picture of the brown creeper, one of my favorite winter birds around here. Great shot he got. You really can appreciate his little kickstand tail and that curved bill. Really cool. And the way the coloring blends in with the bark of the tree. Like they're just really neat birds. So our birding challenge, let me tell you, things are really heating up. We got a lot of close ones there. Strummer Edwards continues to surge into the lead. He's been really acing it this season. But after that, we've really got a tight race. Wayne and myself and Gretchen each have 52 species for the year. Jeannie McCoy, I put her in white. Welcome Jeannie to the leaderboard list. I'm not sure if she got a later start, but she had a fantastic last few weeks. Her last month has been aces, so she caught right up. She's up there with Melissa Mantooth with 51 species. Lucy is doing really well. Steve Tracy and Renee, they all have 50. And Jim Guyton, I left off the Y, typo on Jim's name, I apologize. That was a last minute addition. He just gained another one yesterday. So these are, as of end of day yesterday, they haven't done, it looks like eBird hasn't done their update yet today. So those might be off by 1 or 2, but folks are really getting out there and seeing a lot, which is great. And we're getting into that season where hopefully we'll start adding the migrants that are going to be coming through. So I expect these numbers to surge up again. So as I said, Strummer is in the lead in Mecklenburg. Melissa is in the league in Gaston County. Renee in Cabarrus. Dennis Kent is ahead in Union County. Gretchen in South Carolina, in York. And our own Steve Coggin is in our other category. So keep at it. We're really glad. We're glad to see a new name on the list. And besides these, there really are quite a number of people, similarly placed in the upper 40s who could easily, knock somebody out of contention here. So we're doing good. I'm happy to see that everybody seems to be involved and enjoying it. And fret not, Malia, you're only a few steps behind. You could easily catch up.
Like you say, though, you kind of you just hit a wall,
Yeah, sometimes you plateau. But the season's changing, so I think it's going to go up again.
There's hope.
Congrats to all those leaders.
Okay, we'll quickly do this one. The North Carolina Bird Atlas has gotten cranked up this week, and we could go into details if you want to, Christine, but basically I would just suggest that you go to ncbirdatlas.org. This is something that is done throughout the state every 15 or 20 years, and it will be done for 5 years. So it's a commitment, but very interesting, and there will be an upcoming mass Zoom meeting about how to participate through eBird. So anything else you want to throw in, Christine?
No, not really. It's supposed to be kicking off this month, but I think they're still piecing some bits of it together. But we will have more details soon. So those of you who like citizen science, it'll be great. And you don't need to be an expert by any means. And for those of you who who aren't the type who would be on the previous slide about seeing the most birds, this is more about taking it easy and observing fewer birds more closely for longer periods of time, which is a little different. So it may interest you. So I hope you'll check it out.
Thank you. Okay, our next meeting, I'll just briefly talk about that and then bring it up again at the end to remind you, it'll be Thursday, April 1st. And our speaker is Kate Sutherland, marine biologist with Seabirding Pelagic Trips. So we are looking forward with that, and great photo, Christine. I love that. Very
Thanks.
nice. Okay, now we mentioned a minute ago that we're really, really glad to have our field trips back. So let me, without further ado, call on Steve Coggin to tell us what's up and when's when and where's where.
All right, let me find my slide.
Okay.
Hiding on me.
And thank you, Steve, while Christine's finding her slide for scrambling to get these back on the schedule, because we just found out the end of last week that conditions were changing, and the governor had changed the guidelines, and even though we're going to keep all the same protocols, thanks for scrambling for us, Steve.
Yeah, we have a pretty good schedule coming up, and there'll be more being added to that schedule. And we still have all the rules we had before. We had limit the trip size to 8 people. You have to register with the leader, mask, social distanced, and we'll go to the next one, please. We had one today. Judy ran the first one today, and as she said, everyone loved it. We have another one running on Saturday that unfortunately is full. Judy, did you say the Tuesday one is full also?
Yes, next Tuesday is now full.
And then Chris Bowling will lead one on Sunday the 14th, and I'll do one on March 20th. And March 27th, Ron will lead one at 6 Mile Creek, and we'll probably have some to go into the middle of the week there to fill things out. So this schedule is still a work in progress. And then we have them out all the way to the beginning of May. So there'll be lots of opportunities. We'll probably have some days on weekends where we double up because the demand is going to be really high. So keep checking the website.
Great.
What,
Thank you.
Jeff? Jeff wants to say something.
I'm just going to mention the spring bird count. If y'all want to mark your schedules for April 24th,
Ah,
if y'all are interested, let me know.
Good, thank you for bringing that
Jeff,
up.
I'll go ahead and put that on the calendar.
April 24th, you said, Jeff?
Yes.
Great.
Good. Okay, thank you very much. I'd like to take a second and welcome our newest Mecklenburg Audubon members, and their first names are Becky, Amy, Joelle, Catherine, Andrea, or it might be Andrea, I'm not sure, Kathy, Barry, Kimberly, Jerome, and Tobias. We're so glad to have you as part of our flock, and I think you'll have a whole lot of fun being a Mecklenburg Audubon member. We're still taking board nominations. Janet Palmer, if your mic is unmuted, do you want to say like 4 words about that?
Sure. Hi, I'm Janet Palmer, and I'm chairing the nominating committee with Chris Bowling and Natasha Goins. We are looking for one more nominee for a board position. I'm new to the Mass board this year, but and I'm a member at large. We have a monthly board meeting, except for in July, I believe. We don't typically have a meeting. And there's an August planning meeting. And other than that, it's a commitment. It's typically a 3-year term. You can be as active as you choose to be. There's lots of opportunity. And as you know, we do field trips that we can always use. If you're excited about field trips and want to help with that, we can always use help. And of course, we've got conservation projects going on and advocacy. From what I can see this past year, members are very excited about working for birds. And if you'd like to have fun with us, please get in touch with me. I'll be glad to talk with you, or get in touch with Natasha or Chris and let us know if you would like to be nominated to be on the board with us. And I look forward to talking with someone.
Excellent. Thank you, Janet. Really appreciate that. And just step up. If you want to step up, we'd love to hear from you. All right. So on to our main event, our speaker for tonight. We're eagerly waiting to hear from Larry. Just a reminder, if you have questions as we go along, you can put them in the chat and we will probably address them at the end of his talk. Okay, so I have the great pleasure of introducing Dr. Larry Leamy. He's an accomplished scientist and educator with a really fun sense of humor. Larry received his Bachelor of Science degree in mathematics education from the University of Illinois. He did genetic research and taught a variety of biology courses in California and more recently in the Department of Biological Sciences at the University of North Carolina at Charlotte. And that's where he's now a professor emeritus. Larry has been interested in birds since an early age, and he and his wife Donna are avid birders who especially enjoy trips to see warblers. Oh, my warblers don't show up. Where are you, warblers? Anyway, we all love warblers. So presently, Larry is serving as a board member for Mecklenburg Audubon and is chairman of the education committee. Okay, take it away, Larry.
Okay.
Yay.
Can you all see and hear me?
Yes, sir.
Yes.
Well, first of all, I'd like to thank Malia and all my colleagues on the board for their invitation to give this talk tonight, and for also all the good work they do to keep this great organization going strong. I really appreciate it. I know all the people out there appreciate it as well. So we want to talk about bird hearing here. And I'm going to divide this topic up tonight into 3 parts. Oops. I'm going to start out with how birds hear. You know, the title is How, What, and Why Do Birds Hear? But let's first look at how birds hear. This may be a little, well, let's say, a little tedious at times, but I think it's important that we understand how birds hear before we can figure out why they hear and what they're hearing. So let's take a look first at some background. We all know that birds have ears, of course, but where do these ears come from? Well, I want to go back 150 million years ago. I think most of you know that birds actually evolved from dinosaurs, and specifically from these theropod dinosaurs. Some of you may know that actually the first bird-like reptiles were actually these things that had 4 wings. They've got some wonderful fossils in China where they discovered these 4-winged reptiles. And what had to happen after that is for those back wings to be lost. And then finally we get the 2-winged like birds that we're familiar with today. Here's the classic right here, Archaeopteryx. This was sort of a half reptile, half bird. If you're a paleontologist, you would know what to look for in the bird features and in the reptile features. And here's actually a cast of the actual fossil of Archaeopteryx. So birds evolved from reptiles. By the way, mammals actually evolved before birds, as you can see down here. So mammals were around before birds were. Birds technically belong to the phylum Chordata. Most of you, I think, probably are familiar with the fact that there's a classification system that's in use for all animals, all plants. We divide all the animals in the animal kingdom into a number of what are called phyla, plural, singular phylum. And so in the phylum Chordata, we have the subphylum Vertebrata, named after all those animals that have vertebra in their backbones. And among the vertebrates, we have these 5 groups. You're all familiar with them, but I thought you might be interested in the fact, as you can see from this figure, that birds really only comprise about 17% of all the vertebrates. Fish really make up the bulk. They make up about half of all the vertebrate species. And by the way, the vertebrate species themselves, you might see up here, constitute less than 5% of all the animals. In fact, there are more beetles alone than all the vertebrates of the world. The beetles really rule. In fact, insects really rule if you want to talk about total number of species. Where do ears come from? Well, I mentioned that birds are chordates, and characteristic of chordates is they have a nerve cord and a notochord. The notochord is a stiff rod that the backbone evolves from. That's why they're named chordata. But they also have these gill slits or pharyngeal slits. And you can see the embryos of several different vertebrates here, including a bird right here. Well, the point is, all of these vertebrates, including the human little embryo here, have gill slits at one time or another when they're developing. And these gill slits develop from these arches, and between each arch, there's this cleft. And eventually those push through in fish like this and become these gill slits that we see in fish. But what about terrestrial vertebrates, vertebrates that live on land, including birds? What happens is this: all these pharyngeal arches and clefts and all that tissue in these gills actually becomes the ears and all the associated tissue around the ears. So here's the eardrum, ear canal, and I'll talk about that shortly, but the first pharyngeal pouch, as it's called, actually gives rise to the ears. And that's true in both reptiles and birds as well as in mammals. And in fact, I'll show you this down here. You can see that these pharyngeal areas right here, 1 through 6 is their number here, actually give rise in humans to our outer ear here, our external ear, our pinna as it's called. Well, that's just a little background. Let's take a look now more specifically at bird ears. Bird ears are like our ears, mammal ears, in that they have an outer, middle, and inner part. I'm going to take you through each of these because each of these is important to understand bird hearing. Birds have an ear canal, and that's all they really have for an outer ear. Here's a little diagram here, and here's the ear canal right here. Here's the eardrum inside the ear canal. So the outer ear only consists of the ear canal in birds because birds don't have a pinna like we do. This thing is called a pinna, this external ear out here. Instead, birds just have these holes. That's all they have, is a hole in their side of their heads where the sound wave goes in to affect hearing. Now, that's not quite true. Some owls have what's called a ruff around the edge here, and some of them have a partial covering of that ear called an operculum, or operculi plural. So here's an operculum in an owl ear. But if you look at most birds, they have no external ear at all. They don't have a pinna like we do. And that has ramifications for hearing, as I'll talk to you about. If you look further in here, here's this eardrum. And the eardrum, you're all familiar with the eardrum in humans, separates the outer ear from the middle ear. I want to dwell a little bit on the middle ear because this is a really interesting part of the ear. It's not the key part where hearing occurs. It transmits the vibrations, so it's part of it. We'll get to the key part in a minute here, the inner ear. But this middle ear is this part right here. It's the middle ear cavity. The middle ear in birds has one middle ear ossicle, or one middle ear bone. It's called the stapes or columella. It's actually derived itself from one of the pharyngeal arches that I talked about earlier. What's interesting to me about the middle ear is that birds only have this one bone that transmits the vibration from the eardrum into the inner ear, whereas mammals have 3 middle ear bones. The 3 middle ear bones actually are really interesting in mammals because they consist of the malleus, incus, and stapes. There's that same stapes right there that birds have, but mammals like us have an additional 2 bones, the malleus and the incus. Where those come from is actually came from the skull. And we know that from tracing of cells, which can be done, and also from genetic studies, which I won't worry you with. But the plain fact is, over evolutionary time, mammals, probably because they were selected for finer hearing, actually evolved 3 ear bones or middle ear bones. And as a consequence, mammals can actually hear in many ways better than birds. They have a larger range than most birds, as I'll get into later, as I'll explain later. And so apparently this was important enough in evolution that the development of these 2 extra bones to give better hearing in mammals took place even at an expense. And what was this expense? Well, the expense is this. Here's this middle ear cavity. It's an air-filled cavity, but the tissues in there generally develop a solution that comes right down this, in birds, called what's called the pharyngeal-tympanic tube. We just call it the Eustachian tube, that drains out and clears that middle ear cavity of any infections. Well, you all know, I think, that sometimes among us humans, what happens is we get a middle ear infection. We get a middle ear infection when we have maybe something wrong with our throat or maybe our sinuses. And what happens is this Eustachian tube gets clogged. And so that fluid in the middle ear cavity can't escape out of there. So it gets stuck up there, can't drain, bacteria get in there, and lo and behold, we get a bacterial infection. It's called otitis media. My youngest son, David, when he was a really small child, had a lot of troubles with middle ear infections. And you go to the doctor and you get these antibiotics to try and clear that up. And in some cases, it can really be a problem for certain people. Well, here's what I find interesting. These pharyngeal, tympanic, or Eustachian tubes drain in birds very well. You notice the difference here. Here's a very wide tube in birds. It's actually also situated a little more underneath here. Here on the side, it's really situated more on the side. So this doesn't drain as well. Now, there are little cilia, which are little hair-like projections in the tube itself, in both birds and mammals, that actually beat and get that fluid out of there, or tend to try and get that fluid out of there back into our pharynx or our throat where it drains away. But the problem with mammals is, even though there's cilia in the tube and even some cilia up here in this middle ear cavity, it doesn't always drain very well. And so sometimes we're prone to infections. Well, birds don't even have cilia in the middle ear cavity. They have it in the pharyngeal-tympanic tube, but they don't have it here. Yet they've got such a wide and nicely situated tube here that guess what? They don't get infections. Birds do not get middle ear infections. Mind you, they can get external ear infections, they can get inner ear infections, but they don't get any middle ear infections. So that's a real advantage the birds have. And it's mainly because of this difference in the size of the tubes. Here's something too that I want to talk about next when we talk about the inner ear, before we get to the actual hearing part per se, and that's the part of the inner ear that has to do with balance. It's difficult to talk about the ear and hearing without also talking about these structures in the inner ear that have to do with balance. Balance is actually done in birds by semicircular canals and what are called otolith organs. Let's look at the semicircular canals first. And what it says there when they detect rotational or angular acceleration, what that means is if you move your head side to side like this, or if the bird moves its head side to side like that, or tilts your head like this, that's rotational acceleration or angular acceleration. Here's a generalized diagram of a bird's ear. And these things right up here in purple are the semicircular canals. Some of you have probably heard of these, I'm sure. They're actually located in all of the three basic dimensions of space, the X, Y, Z coordinates, as it were, the horizontal, the anterior, if you will, and what's called the posterior. Notice, there's fluid in these. Notice that there are these little swellings called ampullae at the bases of each of these semicircular canals. What happens is in these ampullae, there are these structures right here called cupolas. These cupolas, there's a gel-like substance here in which there are these little hair cells. What happens is when the head is rotated like that, and so this cupola changes in shape like that, it stimulates these hair cells. And these hair cells fire, or in other words, they transmit an electric message down into this sensory nerve. That sensory nerve goes to the brain, and the brain interprets that and says, aha, you're moving your head side to side, or the bird's moving its head side to side. So that's how birds and us both can instantly figure out how we're moving in space, if we're moving at least in some angular way. You might say, well, how can this be done so fast? Well, literally, this is at the speed, if you will, that electricity travels. It's just instant. It's in a fraction of a second that this transmission from these hair cells takes place to the brain of the bird. All right, so that's part of the balance that birds have in the semicircular canals. And by the way, I should mention, I didn't put this in my slide because we're deep enough into this as it is, I'm sure. But it's only been recently really discovered that birds also have some other canals down in their backs, in their vertebra, that act in a similar way to these semicircular canals and provide them with a really good idea about balance. And birds like this flamingo shown here, for example, actually use that as well for stability or to sense where they are in space and to achieve good balance. That's why flamingos and other birds can stand on one foot like that and do so well. They've got a really good system of balance. And here's the rest of this system of balance. This is very interesting. And by the way, we have exactly the same thing. We have these semicircular canals. We also have what are called otolith organs. Otolith organs. One of them's called the saccule and one of them's called the utricle, or sometimes they're called the utriculus and the sacculus. They detect what's called static balance. Static balance would be if I were a bird, that would be if I were to bend back like that or bend down like that, or the sensation of gravity pulling me down or pulling me straight up, if something was pulling me straight up. That's what these otolith organs do. And how do they do this? Well, here they are down here. Here's the utriculus down here, and here's this little sacculus. Basically, they're little pouches sort of at the base of the semicircular canals. And what they do is as follows. Here again are little hair cells. At the top of these hair cells are what are called otoconia. Otoconia are little calcium carbonate crystals that are embedded. They're embedded in this sort of gel-like matrix with these hair cells. And what happens is when we bend our head forward, or when a bird bends its head forward, these otoconia, which weigh a little more than these hair cells, will bend these hair cells and cause them to fire, or if you will, cause these nerve cells, once again, to transmit these hair cells, which are modified nerve cells, to transmit an impulse to the brain. And it's instantly interpreted, instantly interpreted. I hope none of you have ever had vertigo. I've had several vertigo sessions over the years. If you've ever been lying in bed and all of a sudden turn and feel like the whole world's spinning around you, that's what's called vertigo. Vertigo can happen in humans, it happens in birds. How does it happen? It happens because of these otoconia. Here's what happens. These otoconia, some of them by accident break loose from this gel, and they pass out of typically the utriculus into these semicircular canals. And if they get into the semicircular canals, you see right here, they get in there, they can brush right back by those, and I'll go back a minute, right back by those cupolas that are in the semicircular canals and stimulate the hair cells. Well, meanwhile, the brain is thinking we're moving one way because of what the semicircular canals are saying, because of those otoconia that are shifted out of there. And the utriculus and sacculus think we're doing something else. So the brain is really confused, and it gives this signal that we interpret or that we feel as vertigo. It's an awful sensation. It can actually be corrected by certain maneuvers like the Echolocation maneuver to try and get those little crystals back where they belong. But birds suffer the same thing we do. They can absolutely have vertigo if this happens. Okay, well, let's look now more specifically at the real part of hearing, and that takes place in the other lower region of the inner ear called cochlea. Here's the same diagram again, and in green here you see this cochlea. If you look inside the cochlea, the key structure that is, if you will, the organ of hearing in birds is what's called the basilar papilla. Here's just a rendering of the basilar papilla. It's got a basilar membrane below it and a tectorial membrane above it. And what it really consists of, this middle part right here, is a series, once again, of these little hair cells. They're both tall and short hair cells. And what happens is this basilar membrane vibrates, and actually, when it vibrates, these short and tall hair cells both touch this tectorial membrane, and that causes them once again to what I call fire. It's actually a transmission of their impulses down this nerve and once again to the brain. So they're transmitting an impulse down to the brain. That's what's really happening here. I think a lot of you have probably heard of hair cells in humans. As some of us age, myself included, we sometimes tend to lose hearing. I'd love to be able to hear that little black and white warbler, but I just can't hear very well anymore. That little black and white warbler gives that little high-pitched sound that's become increasingly difficult for me to hear. The reason? I've lost some of these little hair cells. And in humans, they don't grow back. Once you lose the hair cells, they're gone. As more and more of these hair cells die, of course, you begin to lose hearing, and we begin to fit ourselves with hearing aids, etc. Well, of course, birds don't have hearing aids, so what do? Well, I'll show you in a minute. But what happens here for hearing is the following. Let me now take you through the basic process of how hearing occurs. Sound waves come in the opening of the ear in birds. They pass through this external canal here, the auditory canal. They hit the eardrum, and the eardrum vibrates, this tympanic vibrates, and it causes this single bone, the columella, or stapes, to vibrate up against what's called this little round window, and this round window vibrates, and there's fluid inside this cochlea and inside this cochlear duct that continues the impulse or this transmission of the sound wave down into this basilar papilla. And I've already explained this basilar membrane then, which is bathed in this fluid, vibrates, and this vibration causes these little hair cells to hit the tectorial membrane. And once they do that, once they hit on that tectorial membrane, it causes them then to send their impulses to the brain. The brain is where all the sound is sorted out. All these little hair cells do is fire. They send impulses to the brain. The brain is the place where it has to sort all this out. Say, oh, that's a high note, a low note, that's so much loudness, or not so much loudness. Maybe it's a soft note, maybe it's a loud note, maybe it's a higher note, lower note. Maybe it's a musical single note, maybe it's a symphony. The brain sorts all that out. Well, how does the brain detect loudness? Loudness is very simple. Loudness depends upon the number of hair cells that are stimulated. It's very simple. There are thousands of hair cells in these basilar papilla in our ears. The more the hair cells that are stimulated, the louder the sound. So that's how loudness is determined in the brain. But how about frequency? Whether a sound is high or low, whether it's a high-pitched sound like a black and white warbler or a lower-pitched sound. But here's how that's done in birds. Here's a schematic here with the cochlea. And basically what it amounts to is that the hair cells up nearest the round window up here, the oval window and the round window up here, actually produce sounds at higher frequencies, or I should say, they're specifically tuned to vibrate with higher frequency sounds that are then transmitted back, as I've shown you, through the nerve into the brain. As you go further out, down the cochlea, there are lower and lower sounds that basilar membrane responds to. So the basilar membrane responds to different sounds. Here's just a bird that maybe responds to sounds up to 10,000 hertz. Here's a chicken here that responds to sounds from about 0.1 up to 4,000 kilohertz, or 4,000 hertz, I should say. This is K is 1,000. So this is a 10th up to 4,000 hertz. I'll explain what hertz is when we get down to that part too. So that's how frequency is detected. Well, I've already mentioned this business about hair cells. Boy, would I love to have some more hair cells. Hair cells can actually grow from these supporting cells in either of two ways. One way is that these supporting cells just become hair cells. The other way is that supporting hair cells divide and become additional supporting cells and hair cells, either one. Unfortunately, this doesn't occur in us. Once hair cells die, we've had it. But guess what? Birds never go deaf. I don't think I'd want to be a bird because I have a fear of heights. So I don't think I'd like to sit up in some tree way high up. But there's one thing I really envy about birds. They don't go deaf. They grow back their hair cells. As you can imagine, they're all over the world. There are labs trying to figure out how birds and reptiles and some of the other animals are able to grow these hair cells back, whereas mammals have lost this ability. I could get into the little bit of the genetics of that, but let's just say that birds are really lucky in that respect. We're not so lucky. We lose our hair cells, but birds can grow them back. There's one other otolith organ called Legita that I wanted to mention to you. This is a fascinating little part of the cochlea. You can see it right down here at the very end of Legita. In fact, I could go back. I don't want to make too long of backtrack here, but here's Legita right down here. You see this little part right at the very end of the cochlea is another otolith organ called the Legita. So here it is right here. What about the mystery of this? Well, a while back, a few years back, it was discovered that there was some little otoconia in this Legita. That's what makes it an otolith organ. But what was really fascinating is some people thought they were magnetic. It was magnetite. And lo and behold, you can see here, this is May of 2012. People thought, aha, we figured it out. We figured out how pigeons can use magnetic fields to find their way back home. In other words, people thought they had solved the homing pigeon business. The ability of these homing pigeons and other birds maybe to find their way back home. This is 2012. They thought, aha, there are little magnetite otoconia in Legita, and they're using that to sense the magnetic fields in the earth, and they're using these to find their way home. Well, as science often does, a number of other people followed up these experiments done by these investigators. And guess what? In 2019, there's no evidence for any magnetite-based magnoreceptor in the Legita. Further studies showed no, those really weren't magnetite-based otoconial crystals in there. The latest that I have seen is there's a certain protein called Cry4, which is involved in circadian or daily rhythms in all of us, including birds, that actually allows birds to see magnetic fields. So the latest that I have seen is how birds are now being able, they think at least, to sense magnetic fields is from this other protein that actually comes into the eye of birds and allows them actually to see, if you will, quote, see the magnetic fields, and so navigate by those. Again, this isn't hearing, but this was so interesting. And the Legita, of course, is in the ears. And the Legita went from sort of a loser, sort of a winner, I should say, to an absolute loser. Or one of my favorite expressions that might also be good here is chicken one day, feathers the next. So it's not the Legita, apparently, that birds use to navigate. Well, it's one thing to hear sounds, but how about birds locating where that sound comes from? Localizing sounds can be done either in the horizontal plane, that is, if I were to look around in the horizon here, in the left or the right, how do I know where that sound is coming from exactly in this horizontal plane or the azimuth? It's pretty simple. Birds do it the same way we do. If here's a sound over here, let's say on the right side of the head, when that sound is broadcast down on the head, it reaches the right ear if the sound's on the right side quicker than it does the left ear. It also, of course, is going to be a little louder on the right side than on the left side. This is what's called this difference in the time, the interoral. Interoral just means between the two ears, time difference. Or there's the interoral intensity difference. Or just think sound here. So birds do it the same way. If the sound's on the right side of them, they're going to hear it louder in their right ear than the left ear. It's also going to take longer to get to their left ear if the sound is on the right side. That split-second difference, and I mean something like sometimes 30 millionths of a second difference when it reaches the right ear versus the left ear, is enough for the brain to interpret instantly where that sound is coming from. So localizing sound on the horizontal plane is pretty easy. That is, it's well established now in birds that it's done the same way as it is in humans. How about the vertical plane? Elevation, how about below or above a bird? This was a mystery for a long time, and I'll tell you why. In humans, the way we do it is if a sound, let's suppose the sound is coming from our side, it's coming from either a height above our head like this, about even with our ear, or below our ear. Now, if it's coming at the same distance, it's got to be the same distance, so there's got to be an arc here. We're going to actually hear that sound a little differently. It's going to generate a little different spectrum, sound spectrum. Why is that? Because we have this pinna. We have this unusually shaped folded pinna, or what we call ear, that will shape that sound as it comes in from these different heights. And so that difference in the sound spectrum that comes in even a single ear in humans is sufficient for us to be able to locate vertically where that sound is coming from, whether it's coming from below us or above us, and how far above us or how far below us, or right even with us. So in blue here, you sort of see the human using these pinna, or these ears, for localizing sound in the vertical plane. Well, it turns out this was a mystery how birds did it, until a number of experiments were done. People began to realize they use the whole head. The whole head modifies sound. So if sound comes in on one side, even though there's no pinna in most birds to stop the sound and create these different spectra, these are all going to be interpreted about the same on one side. By this time the sound gets to the other side, it's going to be different, because the head shape modifies the sound on the other side of the head. So once again, birds actually use the difference between the two ears in the sound itself, or if you will, what they're calling here the interoral intensity difference. So birds actually make use of both ears. Humans actually could use one ear and detect on the side at least where the sound's coming from in the vertical plane. Owls are even better at this. In the horizontal plane, they do it much like I've already described. I'll go back here again. They do it either of these two ways, except it turns out that actually owls mostly use this time difference. And I'll explain why, rather than this sound difference. So in the horizontal plane, if there's some sound to the right side of an owl, it's going to pick up that difference between the sound It's going to be louder on the right side than it is on the left side. Here's an example right here. Let's say this mouse is over here on this side. You notice that it's going to hear that sound first on this left side. So you can see this interoral time difference. Sound's going to come first to the left ear, then the right ear. It's also going to be, you can see here, louder in the left ear than it is in the right ear. So that makes sense for the horizontal plane. What about the vertical plane? It turns out that owls are really very specially adapted for picking up sound on the vertical plane. And you can imagine why. If they're chasing some mouse that's below them, they want to be able to locate that mouse exactly where it is below them. Well, it turns out they have two ears that are asymmetrical. The left ear is actually a little higher than the right ear, but it's tricky because the left ear is actually pointed downward. The right ear is pointed slightly upward. So sounds below the level of the ears in owls, they hear louder in the left ear. Sounds above the ear are heard louder in the right ear. So even if a mouse or some other prey is right straight in front of an owl, in fact, they can pretty well localize where that mouse is. In fact, they can exactly localize it because of the difference in the sound between those two ears. In other words, they're going to use the interoral intensity difference again. So they use a different method than a lot of the other birds. Turns out the eye placement of owls, notice this barn owl here, they're looking straight ahead of us. The eye placement is in front there, whereas in most birds, of course, it's on the side. So most birds are having to use a different mechanism for localization. And in fact, owls are superior to most other birds that have eyes on the side at spotting or at localizing sounds in front of them. They're much better typically than most of the birds that have eyes on the side. I want to quickly mention there are other adaptations in birds. I don't want to belabor this, but you might ask, how in the world do all these various water birds manage when they dive under the water? Must be awful hard on the ears or the eardrums. Well, you can look at some of these examples of penguins and murres and auks and so forth, cormorants, gannets, and almost all of them have some way to protect their eardrums. So you have a little flap over them, or they have stiffer eardrums, or they have pressure regulators, or one thing or another, so that they're protected underwater. They still hear underwater. They're protected when they dive. They can open these flaps sometimes if they want to hear, if they want to chase something underwater. Okay, so that finishes this first part. I hope that wasn't too tedious, that background information. Now we get to what birds here, and then we'll finally get to why birds here. So let's take a look now at what birds here. Just some basic background on this first. When I talked about the frequency, you can actually show that as a figure in this figure here. If you look at the wave that's generated from the sound, a higher pitch generates more waves in that length of space than it does a lower pitch. Also, as far as and by the way, when we talk about these kinds of figures here, we talk about frequencies, but you and I mostly just talk about the pitch, a higher pitch or a lower pitch. Pitch just describes the frequency. The higher the frequency, the higher the pitch. But we measure them in hertz. Now keep this in mind, I'll emphasize this later. Humans can hear from about 20 hertz to 20,000 hertz. That's the frequency. So we can hear sounds as low as about 20 hertz and as high as about 20,000 hertz. And as far as the figure, and the louder it gets, the higher this wave amplitude gets. Also, of course, you've all heard about decibels, I'm sure. And here's just a little example of how you can measure decibels, how you can measure rather the amount of sound in decibels. Okay, I mentioned that humans hear from about 20 to 20,000 hertz. Any sound higher than 20,000 hertz is called ultrasound. Anything lower than 20 hertz is called infrasound. So this is human-based, but because we hear from about 20 to 20,000 hertz, again, that's back here, we define this ultrasound and infrasound as the sound above or below our range of hearing. Before I tell you how birds or how much birds can hear, or what they're really hearing, how do we measure hearing? Well, you can either anesthetize a poor little puffin like this and put electrodes in it and put a speaker up here and then play that speaker at a certain number of decibels to make sure your sound is constant at different various frequencies. And you see whether those electrodes have responded or not, which is really a response of the auditory nerve. If it understands that it's hearing a sound, it'll say, it'll pass that impulse to the brain. Those electrodes will pick that up. Or you can do stimulus conditioning. This is called auditory evoked potential. This is stimulus conditioning is where you put a bird in some sort of a cage or apparatus like this, and you basically train it to recognize sounds by positive stimulation, which usually means feeding it when it gets the correct sound right, or when it pecks where it's supposed to, for example. I'll show you more specifically an example when we get to it. So these are the two major methods of how sound is measured in animals like birds. I want to mention that many invertebrates can sense vibrations. Even worms can sense vibrations. Or even some of these sea creatures can sense vibrations. Even these things like these cnidarians can sense vibrations. But among the invertebrates, it's mostly insects and only certain specific insects that are actually truly hearing in the sense we think about it. These are mainly the insects that make sounds themselves. So a lot of the animal kingdom, if we take our definition or the way we think about hearing, a lot of the animal kingdom is deaf in the way we think about hearing. But among the vertebrates, I mentioned what those 5 vertebrate classes were. All the vertebrates have balance organs, like I mentioned before. They have what are called mechanoreception structures. They can sense vibrations. And except for fish, they all have a cochlea for hearing. The fish doesn't have a cochlea. So it can sense vibrations, and it can quote, fish can quote hear, but not in the way that the other vertebrates can hear. Well, here's an animal hearing chart with various animals. You see birds are right here. You might think, good grief, birds are just this limited. Well, this chart shows you the major range of bird hearing and frog hearing and so forth. In other words, this is where most of the birds hear the best. This is not the full range of all the birds, as I'll show you. But you can see here, the majority of birds hear best from about 1 to 4,000 hertz in that range. So any sounds from about 1,000 to 4,000 are in their best range for hearing. You can see some of the animals go down into infrasonic and some of them go up here in the ultrasonic frequencies. This is what I mentioned earlier, just a minute ago, that yes, birds may have their greatest sensitivity from about 1 to 4,000 hertz, but take a look at the birds over here. You can see that they vary tremendously. Look at the long-eared owl there. That's quite a range of hearing it has. So it varies. So you can't really say that any given bird hears from 1 to 4,000 hertz, 1,000, 4,000. It varies depending upon the particular species. So again, just to mention, people can hear from 20 to 20,000 hertz. And for a long time, it's assumed that at least most birds can't hear either infrasound or ultrasound. Most birds. However, if that's true, there are certain hummingbirds like these black Jacobins or the blue-throated hummingbird, beautiful little hummingbird, beautiful large hummingbird, that actually sing in the ultrasonic range. Well, if they can sing in the ultrasonic range, in other words, above 20,000 hertz, that suggests surely that they're hearing that sound. Else, why would they be singing it? They've got to be able to hear each other. Our best guess at this point, although I'm going to give you another explanation later, is that maybe we just haven't been able to measure them that well. Maybe they really are hearing that. I'll get to that in another part in just a minute here. Turns out, too, that it's now been discovered that certain birds actually are sensitive to infrasound. Anything below 20 hertz is infrasound. And so here's some examples of birds that are sensitive to infrasound. So at least certain birds can hear in ranges slightly below what we can hear. And I won't worry you with this, but there are all kinds of adaptations for infrasound hearing, including sometimes whole body vibrations that are used. Pigeon actually even has hair cells that respond to infrasounds. You might ask, how about diving birds? How are they hearing underwater? I already mentioned they have all these adaptations for protecting their eardrums underwater. Well, it turns out that you can anesthetize cormorants, or you can put them through training. I won't worry you with the details of this. And however you measure something like a cormorant, this was a great cormorants that were measured underwater. It turns out that underwater they hear exactly almost the same as above water, which really actually means they hear better underwater. And they hear better underwater because sound doesn't travel as well underwater as it does in the air. So that means actually they're better underwater than they are above water. So some of these birds, cormorants especially, have actually been selected for better underwater hearing than above water hearing, or in air hearing. So what are birds hearing? Well, we have what's called relative pitch. We can hear one tone, and then if we sing a song, for instance, in one octave, we can recognize it in another octave. If we sing happy birthday to somebody, a happy birthday song, in one octave, we sing it in a higher octave, we recognize it instantly. Birds can't. They have absolute pitch. They depend upon a certain pitch. And if you sang happy birthday, or they sang their own song in one octave or a certain pitch, if you modify that pitch, they don't recognize it. So for a long time, it was thought that birds just simply have absolute pitch for song recognition. But what about timbre? Timbre is the difference when you have sounds that are all the same pitch, but they come from different instruments. For example, a tuning fork or a flute or our voice or a violin. You all know that the same note played on a flute sounds different than played on a violin or an oboe or any other instrument, or our voice. That difference is what's called timbre. And it's long been known that birds seem to be able to recognize timbre. So what do birds actually use? Well, I want to take you through a very important experiment that shows us what birds really use to recognize sounds, or what kinds of sounds, I should say, they recognize. In this experiment, they used 5 starlings, and they trained them. When they pecked this middle hole, they got a little start of a little vocal recording here. And what they tried to get them to recognize was either an ascending sequence of 4 notes or a descending. If they had an ascending, they had to peck the left port here for correctness, descending the right port for correctness. So that's what they did with these starlings. They trained them, and you can see they started out about 50% at random, and they got so that they got up to about 90% correct in their responses. There's an example of what 4 of these notes sounded like in the ascending sequence. There's just one example. They used all sorts of different tones and sequences here. So the starlings were well trained. So then what they do? Here's what they did next. Now, bear with me on this. I know this looks complicated. Here's what they did next. They decided, all right, we've got them trained. Now we're going to try and confuse them. We're going to change the pitch. I'm going to keep the timbre, change the pitch, and see what happens. That's depicted there. The blue here is are the train. The red is the difference in when they change the pitch. So here's the train bird, and here's the change pitch. You notice that last pitch there was lower than the first one. So what happened? Well, there's the train ones up there above. Once they changed the pitch, they got down to about 50%. In other words, they picked the left or right sides at random. They didn't learn. They were confused. This suggests maybe the absolute pitch is the main thing they're using for sound recognition. But the next step, they said, all right, well, if that's true, now let's try changing the timbre, but keeping the pitch and see what happens. Here's the trained again. Here's the change timbre. Notice that change timbre, that's just a piano note. I'll do this again. Here's the piano. Well, once again, they don't show the train ones up there. Once again, it's down near 50%. It totally fooled them. So what's this say? This says that birds can't recognize songs that they're singing or other cohorts of them, of their species, are singing. Just from the pitch alone, they're obviously confused if you change the timbre. So they did a final experiment, and this is the key experiment. Here's the train, here's the changed pattern again for timbre, and then they did what's called vocoded changes. They changed the whole spectral pattern, as it's called. There's a little figure down here. I don't know if you can make much sense of this, but the blue is the trained birds, and the red is how they changed the spectrum. They're keeping the pitch, but they changed the whole spectral pattern. So here's the train. Here's changing of timbre, and here's vocoded. That to us just sounds like a whisper. But guess what? The birds recognize this vocoded thing when they couldn't recognize this changed timbre pattern. That whispering effect, they were able actually to recognize that better than they were, that's the red part here, better than they were the changed pattern of timbre. So all this suggests what? This suggests that birds don't just use pitch, they don't just use timbre, they use a combination of both. And you can think of this as what's called the overall spectral shape. Here's one way to think of this. Here's a white-crowned sparrow song, a red-winged blackbird. The whole spectral shape is shown right here. That's what they're recognizing. And that spectral shape is a combination of both timbre and pitch and different waves throughout that song. Okay, so that's a little bit on what birds are hearing. I think why birds hear we're all probably familiar with. If a bird can't hear a song of its own flock, it's in trouble, obviously. Birds are very vocal. They've got to have good hearing. Maybe that's why selection favored the retention of their hair cells and the replacement of their hair cells if they lost them, so they wouldn't lose hearing at all, never become deaf. So they obviously have to rely on their hearing. And here are just some of the categories that I think are important in showing how birds need that hearing. How do they need it? Why do they need it? Well, they've got to be able to communicate with other birds about what? Mating and establishing territories, about predators that may be coming after them. Maybe they need it to detect food sources, or maybe they need it for navigation, or maybe they even sense sound and avoid it, what we would call noise pollution. Let's look at each of those in turn. How about songs? Most birds tend to learn their songs from their parents, often their fathers. And it's been shown over and over again, if their fathers aren't very good or produce abnormal songs, or I love this, or negligent, they're out with the boys, they're offspring do not produce sounds that are that good. Their song is hampered. If you also only have chicks raised by sounds from a, let's say, a recording, they don't do as well either. Now, there's some exceptions. There was a recent study done that showed that early life stress, which typically results in poor sounds, in some cases, in these little rice finches in which the study was done, actually produced songs with increased complexity, as you can see from this little figure down here. That's a little odd because actually females generally prefer males that can sing more complex songs. So this seems to be opposite of what you'd expect. Some more work needs to be done here, but I can tell you in general, the literature suggests that stresses early on produce poor songs in birds. How about these hummingbirds I mentioned earlier? Well, we now know that those birds probably are hearing ultrasonic sounds. How and why? Because they're living out in really noisy places. They live out in places where there are waterfowl falls and other bird songs and wind and you name it. And so probably they've been selected to sing songs in an ultrasonic range because of their noisy home habitats. That's the best hypothesis out there at the moment. You all know too, of course, as birders, that bird songs can be songs, they can be notes. And I'm going to play for you a couple of a song or two and a note from an eastern towhee just to show you what birds have to be able to pick up on. Here's the typical drink your tea song of a towhee. You've all heard that. Now here's a call that you've all heard, which is more of an alarm call for a towhee. And then here's a flight call. So the point is this, here are birds making all these calls and notes. Clearly, if another bird can't hear an alarm call, it's in trouble. So hearing is paramount to birds. They have got to be able to hear these calls. One of the things I found interesting that I did not know is that some birds actually communicate with their unborn chicks. They have found that in these little rice finches, zebra finches, excuse me. Turns out that these zebra finches, if it's really hot out, sing a slightly different song to communicate with the baby bird in the egg, in the shell, and that induces that baby bird to hatch earlier. And it comes out, if it comes out earlier, it's a little smaller and it's able to cope with the hot weather better. You might think, oh, a smaller bird wouldn't fare as well, but they've done studies to show, in fact, those smaller birds actually fare quite well. Now, the bird is obviously not thinking, oh, I've got to speed up my metabolism and get out of this egg. It's clearly some sort of hormonal cue that this song elicits, but that actually happens. You also may know that lots of ducks and geese speak to their eggs. That is, they're singing, and that song is already learned by those chicks before they hatch out. They know their mother's song. I showed you those owls before, which had eyes in the front of their head. That's typical for a predator. But prey species mostly have eyes on the sides of their heads, and evolutionarily that came about because, of course, they want to be able to see as wide a field as possible to look for predators. Well, so the birds that have eyes on the sides of their heads, they match up with pretty much their ears on the sides of the head. So birds, you can use both their eyes and their ears in the same plane, at least those that have them on the sides of their heads. So there's a lot of literature that suggests birds use a combination of hearing and vision. But what about when a robin like this tilts its head? I think I was always told that a bird hears these worms and it's hearing those, and then once it hears them, it spots them and picks them up. Well, all the studies that have been done, including studies that have actually used low frequency hearing, birds didn't pay any attention to it at all. People have drilled holes in their ground and stuffed worms in them and lower so they couldn't see them. They didn't pay any attention, but they left a little part of them out. They saw them and picked them out. They coated worms with all sorts of nasty stuff. They don't care about their smell. So it's mostly all about hearing. There might be some, all about sight, excuse me. There might be some hearing involved, but for the most part, birds are tilting their ears because their heads, because their eyes are on the sides of their heads. They've got to tilt them to see what maybe they first heard, perhaps, but to get a better sight on some worm that they can then go for. So birds are mostly tilting their heads because they want to see in the direction they're looking at, maybe some worm that they want to go after. You all know, of course, vocalization is important to attract mates. So obviously, if a bird can't hear a song, let's say a female can't hear a song from a male, mating is not generally going to take place. And it turns out sexual selection has favored females that prefer large or complicated song repertoires. I got to play you this one because this is the world record loudest call, this bellbird. Oops. A little bit about woodpeckers. Woodpeckers can hear at quite low frequencies, and they use that hearing to detect insects underneath the bark. They can peck on hollow places sometimes, and hollow places are good for insects that are in that bark. So they're tapping often to detect hollow places where there might be insects. They often get confused on houses too, because electrical wires and other utilities in houses make sounds that they sometimes think are insects. And this is why they wind up pecking on houses. I talked about owls earlier, and owls are tremendously adapted to locate sound. And here are some of the examples of how this some of these adaptations. But for hearing, they have this wonderful facial ruff. As already mentioned, they have these unequal ears and some of these other characteristics that make them tremendously good at locating prey, especially in front of them. And I want to also mention echolocation. There are some birds, swiftlets and oil birds, here's an oil bird right here, that can actually echolocate. So they're putting out little chirps, sort of like bats. They're not as good as bats. They're more crude, but they can echolocate because they often live in caves and they don't want to run into each other. And so this is a good way to locate flying insects and other objects in the air, including other oil birds or swiftlets. It turns out that some penguins and maybe some other water birds, diving birds specifically that we don't know about, may also be echolocating. Certainly, we now know that some of these diving birds are using their hearing for locating prey underneath the water. But some of them also may be echolocating to locate prey under the water. I've already mentioned pigeons earlier, how it was thought they might be using that Legita, well, it turns out maybe instead they're using their infrasound capability to find their way home. Maybe, and this is speculative still, maybe these very low frequency sounds brought about by what, ocean waves in the distance, all sorts of geological low frequency sounds that we can't even imagine, are what's giving these pigeons a roadmap back home. It's possible. That's at least one hypothesis out there. And finally, I want to wind up with this before I go to the conclusion. There's a lot of increasing literature is coming out now about sound pollution, noise pollution. We now know that certain birds sing differently if they're in urban areas than they do in other areas. We now know that noise is just a really lot of noise is not good for birds. They don't nest as frequently. They don't stay around as frequently, or if they do, they often suffer in reproduction. And so clearly noise pollution is a factor hampering bird populations. I want to conclude now with just some quickies about how birds hear. Here are the major sorts of things I talked about how birds hear. I talked about the sensory papilla and how they get vibrations through that inner ear and they're transmitted those impulses to the brain where it's sorted out. They've got wonderful adaptations for localization and hearing in air and in water. Wonderfully, they don't get middle ear infections and they don't go deaf. They're varyingly sensitive to different frequencies of sound. Some can even detect ultrasound or infrasound. And they clearly rely on both pitch and timbre and the whole spectral shape to recognize songs. And why birds hear, of course, I've just covered that. Had to do with territories, predators, and so forth. Now here's a break for you all coming up. Okay, now we're done with all this stuff. Donna and I love warblers, and so Donna actually, she told me not to tell you this, but she said, put a little warbler up in most of those slides. If they aren't paying any attention or if they don't care anything about this, at least they'll look at a warbler. So here are the what have I got here? 31, 1, 2, 3, 4, 5, 6, 18, and I put in 18 warblers in some of these slides. So I kind of fooled you here. I've got some Western ones in here too. I've yet to see a couple of these like the red-faced I haven't seen yet. But here are the warblers that you may have seen as we went through these slides. And I hope those of you who are birders and love warblers like I do, that you recognize most of these. Be happy to take questions. And thank you for your participation and interest.
Thank you so much, Larry. That was just fascinating. My head is exploding. I learned so much.
Mine too,
yeah, we do have a few questions in the chat. Let's see about this Legita. Did they ever figure out? You talked about what they thought it was, its function was, but it wasn't. Is there any other? Steve Jenkins was asking, is there anything? Do they know what the function of the Legita is?
Legita in lower vertebrates is actually used in part for hearing and actually for mechanoreception, but it now looks like it's a big nothing in birds. It looks like those crystals were just there by chance, by accident, in fact. So it's now thought that it doesn't have any function, or maybe it helps a little in vertical localization, that's all. Maybe a little bit in localization in the vertical plane, but otherwise no known function for the Legita.
Good, good. Steve, Doc Jenkins, you had some others. Is there a predictable pattern to hair cell loss? You want to unmute yourself and ask that one if you're still on?
Yeah, I was just curious if, based on the location of the hair cells in the inner ear or the specific frequencies that they were assigned to, is there a predictable pattern with which hair cells will be lost?
In humans, you're talking about.
Well, humans or any creatures.
Well, they're replaced. Oh, okay. I suppose that birds lose them too. I'm not sure what the pattern of loss there might be, but there have been a lot of studies of humans and hair loss in humans, obviously, because we don't replace them. And what often happens with age is the higher frequency hair cells are lost. And I don't think I explained this very well, but different parts of the basilar membrane are responsive to different frequencies.
Right.
And the hair cells up closest to the middle ear respond to higher frequencies. And those, unfortunately, are often the ones that are lost first.
So are they just physically disrupted more? Or?
It is, and they're also just plain aging in some people. There are also genetic predisposition for hair loss in some people. So both genetic and environmental, just like everything else.
Okay, good. Thanks.
Sure.
I also see that Angie asked, what mechanism do mimics and talking birds use to learn new phrases?
Those are some of the smarter birds in the bird world. They have actually a higher brain. They have a larger brain in the medulla, which is the hindbrain, where all the auditory impulses come. By the way, the owl hindbrain, the medulla, as it's called, is also large compared to a lot of the other birds. And they use those extra neurons in that medulla back in that hindbrain to be able to mimic. In other words, they have that peculiar ability to mimic because they have a different construction of the hindbrain.
Interesting. Very interesting. I noticed the other day, Susan Ellerman posted on Mech Birds that she was, I guess, in line for a vaccine at a school, and there was a mockingbird that was doing a perfect imitation of the school fire bell. And I was curious about whether, if there were people in the parking lot, which would be something that would happen during a fire drill, was the mockingbird making any visual and auditory, putting the two images and sounds together, or is that not something that's feasible?
I can't answer that one, Malia. I have no idea on that one. By the way, I should mention something else for that previous question. Certain drugs like streptomycin, a lot of the mycin family, they can wipe out hair cells.
Woo.
In fact, that happened to my mother-in-law when she had spinal meningitis. They recovered her with a real potpourri of mycin drugs, but she lost almost all of her hearing as a consequence. And so that's all, certain of those drugs really wipe out the hair cells.
Wow.
Sorry, that was an aside.
James also asked or made a comment that he was always told in his shop that it was not how loud the noises were, but how constant the frequencies were, like hearing it for a long period of time. That really caused more hearing problems, hearing the same thing over and over and over and over and over for a long period of time.
Yeah, consistency is important, although they've documented in humans, of course, that hearing for people that are working in really high intensity places like, a jackhammer or a big band or something, that will really ruin your hair cells. And I'm sure it's the same for birds. Birds that are subject to really loud noises are subject to lose their hair cells. They can regenerate them, but they can lose them. But consistency is also important.
And I see what, go ahead, Christine.
I just have one question. Someone from Facebook wanted to know if you could make the slides available. I know sometimes we do that when we post the video on our website.
Sure.
Okay, great. I will let them know to look for it. Judy.
Now, mind you, we don't have all these detailed, wonderful explanations in there.
Well, she can look through them while she's listening to the recording again.
Oh, okay. Sure, be happy to.
Thanks, Larry.
And then one more I see from CDOC is, what advantage do 3 auditory ossicles have over 1 ossicle?
They actually give us not only an expanded range of hearing. You notice most birds don't hear from 20 to 20,000. They actually give us finer hearing. Birds actually have pretty good hearing within their range, but we have a finer hearing throughout our range than birds do. In fact, we're paying the penalty, I think, of that otitis media because of the construction of that middle ear with those 3 ear ossicles. That's what everybody seems to think in the literature.
Did you, there's another Bill Anderson asked,
Anderson.
okay, why do certain birds like parrots and mockingbirds copy sounds? Is that brain or is that hearing? I mean, like mimics and stuff like that.
I think that group, and this is partly speculative. I'm not sure I entirely know the answer, but my best guess would be that those birds in that group that are mimickers, there are a bunch of them, probably evolutionarily were selected to mimic because it helped them in some way. And I suspect how it helped them is they recognized each other's songs and began to mimic them, and that's the way they evolved. And now they continue to mimic any sound that they hear. That's my guess, and it's just a guess, but that wouldn't be around, that mimicking ability, if it weren't selectively important in some way or another. That would be my best guess as to why it might be important. Does that make sense?
Yeah, to me. I don't know if it does to Bill.
Well,
William.
not everything is selected. I mean, there are things that are left over from earlier selection processes that don't necessarily, you can't pinpoint necessarily to some advantage. But I suspect there must have been some evolutionary advantage at one point or another to be able to mimic a sound.
William says yes, that does help.
Okay, well, Larry, I know we've worn you out, but that was just, again, just fascinating and an excellent detailed presentation. You know, people are making comments that they can see why you, well, you know, they wish they had been in your classes because it was really, really good. Thank you so much.
Malia, I made all this up.
You did? Well,
Yeah.
you fooled me.
Oh, okay.
You delivered it with such a plum that we
Oh, I see.
we all absorbed it.
I appreciate it. Thank
You're
you.
welcome. Thank you so much for your time. All right, well, we'll get ready and wrap it up, but I do want to invite everyone to come to our meeting on April 1st. And again, our speaker is Kate Sutherland. She's a marine biologist, a boat co-captain, a deckhand, and a naturalist guide who leads pelagic trips for the company Seabirding off of the Hatteras coast. So I know she's got some phenomenal stories and probably some great pictures. So we look forward to seeing all of you who are leaving us now on Facebook Live on April 1st. So good night to all of you and good birding.
Thank you, Malia.